Hello, my name is Judit Garcia Gonzalez, and I'm a postdoctoral fellow at the Icahn School of Medicine at Mount Sinai. Today, I will walk you through how to run pathway-specific polygenic risk scores, a tool that has been recently developed in the Polygenic Lab, led by Paul O'Reilly. So first of all, it's important to know what are and why it's interesting to use pathway polygenic risk scores. And for that, I'm going to use one example that most of you might be familiar with, which is the PRSet GWAS for schizophrenia. So we know that psychiatric disorders like schizophrenia have complex etiologies where different environmental and genetic factors contribute to the liability of disease. When we talk about genetic factors, we can imagine that GWAS is a composite of signals where each signal might represent functional routes to disease. For example, there might be some genomic regions that harbor genetic risks associated with abnormal synaptic pruning. There might be other regions that are associated with biological pathways and processes related to immune activation, and other regions in the genome that pick up signals associated with cannabis consumption. So the idea behind pathway-specific polygenic scores is that instead of aggregating effects across the entire genome, we will aggregate them across relevant pathways. And because we are separating the genetic contribution of a trait accounting for the genomic substructure, we hope that pathway polygenic scores might be more useful for patient stratification or to investigate the disease heterogeneity. Because now one single individual, instead of having one single polygenic score, will have as many scores as pathways we investigate. So to calculate these pathway scores, I will walk you through the tool PRSet, which is an extension of the PRECISE software, which uses a clamping and p-value thresholding method. And it's important to know that the pathways that can be any type of gene set that reflects the encoding of different biological functions. So this tool is quite flexible to define pathways. PRECISE, as well as the newest version of PRECISE, have been developed by Dr. Sam Choi. And you can find the software and the manual in this website, precise.info. And there in the website, you will find a quick start, how to download it, and also some more detailed guide about what are the available commands and what is the method, how the method works. So how to use PRECISE? PRECISE is very similar to PRECISE in terms of use, where we need as an input the GWAS summary statistics and individual level data for genotype and phenotype. But for PRECISE, we also require information about the pathways or the gene sets that we want to use. As I say, the tool is quite flexible. Pathways can be defined in a variety of ways, including pathways defined by existing canonical databases like GeneOntology or Reactome, but also by experimental perturbations or some functional outputs like gene co-expression or protein-protein interactions. So I'll go in a bit more detail on the different input options that are available to define pathways. One is using GMT and GTF files, using the commands mseq, db, and GTF. The commands that are for PRECISE will be shown in green. So mseq will contain the information related to pathways. So that is the genes that form each pathway. And you will need a file where each pathway is specified in a different row with all the genes that compose that pathway. Then the GTF file will contain the position coordinates for each gene. So in general, GTF files contain different features. And by default, PRECISE uses the features exon, gene, protein coding, or CDS. But this can be modified with the command feature. The second option that can be used is a bed file. In this case, you will need to include one bed file per pathway. So if you want to include multiple pathways, then bed files can be included separated by a comma. In the bed file, 3 columns are required, and those are chromosome name, start position, and end position. One important thing to know when using bed files is that they are indexed differently as PLINK files. So whereas for PLINK files are 1 base, the bed files are 0 base. So you can see in this example that we have a sequence of nucleotides, and whereas for the 1 base indexing, 1 nucleotide is 1 position, the 0 base indexing will use a range around each nucleotide. So you will need to subtract -1 in the 0 base indexing for the bed files. The input option 3 is an SNP list, so similar as the bed files, each pathway will be a different file and they can be separated by comma as in this example here. So when you download PRECISE or PRESET, there is some toy data that you can use. And this is the commands that you can use, like you define the keywords, your target sample. In this case, I'm using the first option to define the pathways. Then you can set the number of permutations that you want to run, and also the name of the output. So after walking you through the input, we can see step by step how PRESET calculates the pathway polygenic scores. To do that, I will compare it with the genome-wide PRS. So whereas for the genome-wide PRS, you do clamping and p-value thresholding for a given R-square and linkage disequilibrium, for pathway PRS, this will be done for each pathway separately. Then, similarly, whereas for the genome-wide PRS, each individual will have only 1 polygenic score. For pathway PRS, each individual will have K scores for K number of pathways. And for the results, whereas for genome-wide PRS, what is reported is the association measure, which is like the phenotype variance explained by the genome-wide PRS. In the case of the pathway scores, there will be the same, the association of phenotype variance explained by each pathway PRS, but you can also report enrichment of GWAS signal in the pathway, which is based on the R-squares of the pathway score. So I'll go a bit in more detail about these 2 different measures that PRESET can output. So when we are talking about association, what PRESET will output is the self-contained p-value. So it's just the regression of the phenotype on pathway PRS and covariates. So pathways that have many genes containing SNPs that are associated with the phenotype are highlighted here in pink, and those will be significant. But it's important to note that for these results, the self-contained p-value does not account for pathway size. And for example, a big pathway will be more likely to be significant because it's easier to have a larger number of SNPs associated with the phenotype. But the second output that we can have is this enrichment that will be output as competitive p-value. This is resulting from a permutation procedure, and this will test whether a pathway is more associated with the phenotype compared to null pathways that have the same size. So in this case, with the competitive p-value, we'll be accounting for the size of each pathway. In this plot, I'll illustrate how we calculate this competitive p-value that accounts for the pathway size. We can imagine a pathway A that has some SNPs in the unique region of the pathway, and for which we will calculate an observed p-value. Then we will calculate as many null pathways as permutations we run, and these null pathways will have the same number of post-clamped SNPs as pathway A, but they will have been, these SNPs will be randomly allocated. So we will obtain a distribution of null p-values, because each pathway will give us a p-value for association. And the competitive p-value will be defined as the number of tests under the node that have a p-value smaller than our observed p-value plus 1, divided by the number of tests performed plus 1. So this is how the competitive p-values are calculated. This approach and this output is similar to the type of enrichment analysis that are performed by MAGMA and LD score regression, but pathway PRS have the advantage that they provide individual level data that can be used for other applications. So let's see how the output of PRSet looks like. Similar to Precise, for every PRSet run, we will have a log file with extension .log, and this will contain the commands used for the analysis and all the information regarding the filtering, the fields that were selected, etc. Then we will have another file with extension Precise, and this will contain the polygenic model for each pathway across thresholds. So in this case, I was running only the threshold 1, which contains all the SNPs, but you can see that there is information for each pathway, the R-squared, the self-contained p-value, coefficient for the regression, standard error, and number of SNPs. You will always see this base pathway, which is the background pathway that is used for the competitive p-value calculation. Then we have the best file, which will give us scores for each individual and each pathway. So here, each row will be one individual, and then the score for each pathway will be indicated in a separate column. So there will be as many columns as pathways you are using. In this case, we see 4 examples from the KEGG database. If you wanted all the scores at all the p-value thresholds, because this best file will report only the one with the best p-value threshold, you can use the command all. Finally, we will have another file with the extension summary that will have the information of the best model fit of each phenotype pathway. And in this case, each pathway will be a row, and then it will contain, again, information on the R-squared, null p-value, coefficient, and also this competitive p-value that I was talking about that will indicate the enrichment of this pathway compared to other pathways of similar size. Finally, I would like to give some heads up and useful commands. So for example, some commands that might be useful for the user is the window size, which is like, for example, if you are defining SNPs that are within a gene, you can extend that window. And so you can include some number n bases to the 3' region of the region, or to the 5' region of each gene. You can also exclude SNPs that are within a certain range with the command xrange. So for example, the MHC complex or the APOE region, if you want to do some Alzheimer's analysis and you want to exclude that one. If you want to improve the computational efficiency of PRSet, you can parallelize the process using the thread command. You can also speed up a little bit PRSet at the expense of increasing the memory using the ultra command. And you can also use the keep and the extract commands to exclude individuals or SNPs from the analysis. If you are doing permutations to calculate the competitive p-values, this can be computationally intensive. So it might be useful to adjust for the covariates like sex or age beforehand on your phenotype, and then use the residuals as the phenotype, because this will speed up the process. So this was this quick introduction to PRSet. Thank you for listening. If there is anything that is not clear or you have any questions, there is a Q&A session the 15th of October at 11:30. And this is the Zoom meeting ID. So I'll be happy to clarify or answer any questions. Thank you.